Among the many strategies emerging in anti-aging science, one of the most promising involves the restoration of NAD levels in aging cells, and at the heart of this lies vitamin B3, or niacin. As a precursor to NAD, nicotinamide adenine dinucleotide, niacin plays a central role in the maintenance of cellular metabolism, mitochondrial function, DNA repair, and, most importantly, for aging autophagy. The decline of NAD with age is now recognized as a key factor driving cellular dysfunction, energy failure, and the gradual breakdown of proteostasis. Restoring NAD levels through niacin or its advanced derivatives like NMN, nicotinamide mononucleotide, has emerged as a compelling strategy to not only activate autophagy, but to make it more precise, sustained, and protective in older cells. NAD is a universal coenzyme required for hundreds of enzymatic reactions, especially those involved in redox metabolism and cell signaling. But its most critical anti-aging functions are linked to the activation of sirtuins, particularly SIRT1, a class of NAD-dependent deacetylases that regulate stress responses, mitochondrial biogenesis, DNA repair, and autophagy. When NAD levels drop, sirtuin activity declines sharply, impairing the cell's ability to respond to stress and triggering the accumulation of damaged proteins and organelles. This leads to a backlog of cellular debris and mitochondrial failure, conditions that autophagy is supposed to counteract, but often cannot in a low NAD environment. This is where niacin becomes not just a vitamin, but a therapeutic tool. Through the salvage pathway, niacin and its related compounds, nicotinamide riboside, NR, NMN, and nicotinic acid, can replenish the cellular pool of NAD. NMN in particular has attracted attention because it bypasses several rate-limiting steps in NAD synthesis and appears to be highly bioavailable in mammalian systems. By raising intracellular NAD levels, NMN and niacin effectively reactivate SIRT1, triggering a cascade of downstream effects that directly influence autophagy. SIRT1 promotes autophagy by deacetylating essential autophagy-related proteins ATG5, ATG7, and LC3, as well as FOXO transcription factors and PGC1A, which enhance mitochondrial quality control. This deacetylation increases autophagic flux, the rate at which damaged components are identified, encapsulated, and degraded in lysosomes. In aging cells where autophagy is often stalled at various stages, NAD restoration reinitiates this process, clearing damaged mitochondria, suppressing inflammation, and restoring energy balance. Importantly, this rejuvenation is not just theoretical. Animal studies have shown that NMN supplementation improves insulin sensitivity, reverses age-related arterial stiffening, and enhances endurance, all outcomes tied to improved mitochondrial and autophagic health. Beyond SIRT1, NAD also supports the function of PARPs, poly-ADP ribose polymerases, which are activated in response to DNA damage. While chronic PRP activation can deplete NAD, adequate niacin intake ensures that both DNA repair and sirtuin activity are sustained. This dual function, maintaining genome stability while enabling efficient cellular cleanup, highlights why NAD balance is so central to aging biology. Another important aspect is the metabolic environment in which NAD operates. Low carbohydrate or fasting states naturally raise the NAD/NADH ratio, favoring oxidative metabolism and enhancing SIRT1 activity. But in older individuals or those with metabolic syndrome, these pathways can be blunted due to chronically low NAD levels. Niacin supplementation, particularly in the form of NMN or NR, can mimic the NAD surge typically seen during fasting, thereby promoting autophagy even in individuals who cannot adhere to strict caloric restriction protocols. In essence, NAD precursors may serve as fasting emetics, activating many of the same longevity pathways without requiring prolonged nutrient deprivation. Of course, not all forms of vitamin B3 are equal in this context. While nicotinic acid and nicotinamide, the traditional B3 vitamins, do raise NAD levels, they can also inhibit sirtuins at higher doses due to product feedback inhibition. NMN and NR avoid this problem by more efficiently entering the NAD salvage pathway, supporting sustained sirtuin activation without inhibition. This makes them more suitable for therapeutic supplementation, especially when targeting autophagy and aging. Long-term safety data on NMN is still accumulating, But so far, results from both animal and early human studies suggest a high degree of tolerance, especially at doses up to 500 milligrams per day. Moreover, NMN appears to cross the blood-brain barrier and may support neuronal autophagy as well, a critical feature given the role of defective autophagy in neurodegenerative diseases like Alzheimer's and Parkinson's. The broader implication is that autophagy is not simply a stress response. It's a regulated, energy-intensive process that depends on adequate nutrient sensing, enzymatic activation, and mitochondrial readiness. NAD sits at the center of all three. With age, these systems become decoupled, leading to partial autophagy or stalled cleanup processes that contribute to cellular senescence and chronic inflammation. Niacin, and especially NMN, represents a way to restore this integration, reactivating the machinery of autophagy in a way that aligns with a cell's deeper metabolic needs. In conclusion, vitamin B3, long thought of as just another water-soluble nutrient, has proven itself to be a linchpin in the biochemistry of aging. Through its role in NAD biosynthesis, it controls the activation of sirtuins, the efficiency of autophagy, and the overall resilience of the cell to oxidative and metabolic stress. Supplementation with niacin or its advanced derivatives like NMN offers a promising path to restoring youthful autophagic function without the harsh demands of long-term fasting. It brings precision, sustainability, and energy back to the cellular cleanup systems that keep us young. In the growing toolkit of anti-aging interventions, NAD restoration stands as both a fundamental strategy and a gateway to deeper regenerative potential.